Great.
Good afternoon, everybody, and welcome to Psychiatry Grand Rounds. Just a couple of announcements. Grand Rounds will be continued to be in person and on Zoom. And if you would like a lunch, please RSVP to Karen Lambert. To receive credit for attending today's lecture, physicians, please go to the Clouds CME. Everyone else should complete the evaluation using the link that came with the Grand Rounds announcement from Karen Lambert this past Monday. And also, if you're here in person, please sign in. If anyone needs touchdown space in an office, either before or after Grand Rounds, please contact Diane George. And for those of you on Zoom today, please type questions for the speaker in the chat. Now, next week, there will be no Grand Rounds because of the ACNP meeting. The next Grand Rounds will be in 2 weeks on December 19th here in Amphitheater 1. That will be the last Grand Rounds in Amphitheater 1. In January, we will be back in the Lazar Auditorium. The speaker in 2 weeks on December 19th is Dr. Alexander Lichtenberg, who's the chief resident in neuropsychiatry. And the title of his talk is Phenomenological and Neurological Explanations of Psychosis, Attempting a Synthesis. It's my pleasure today to introduce one of our faculty, one of our newer faculty, Dr. Ryan Logan, who is a professor of psychiatry and neurobiology here at UMass Chan. Dr. Logan graduated from the University of Maine with a doctorate in biomedical sciences in 2009. He then completed postdoctoral fellowships at Rutgers University in immunology and at the Jackson Laboratory in bioinformatics, and at the University of Pittsburgh in molecular neuroscience. He was an assistant professor in psychiatry at the University of Pittsburgh School of Medicine from 2015 to 2020, and then an associate professor in pharmacology at BU School of Medicine from 2021 to 2023. As you will see from today's talk, his work focuses on understanding the mechanisms underlying the relationships between sleep, circadian rhythms, and psychiatric disorders and addiction disorders, with the goal of developing novel therapeutics for the treatment of mood and substance misuse disorders. His work has been funded by NIDA and NIAAA, and he has received many, many grants, and he's published in some of our most prestigious journals. Dr. Logan has won many awards. I'll just mention a few. A recent one here at UMass was the Dan and Diane Riccio Fund for Neuroscience Research. And he is also an elected associate member in the American College of Neuropsychopharmacology. The title of Dr. Logan's talk is Sleep and Circadian Rhythm Dysfunction and Opioid Addiction.
Thank you, Dr. Rothschild, for the introduction. I'm glad to be able to finally do this. So I'm coming up on a 2-year anniversary of coming to UMass. I was supposed to do Grand Rounds last year, but ran into some interesting health issues. So I'm going to talk kind of a broad overview of what our lab has been focused on over really the past 5 or so years. And what we've been mainly interested in is how sleep and circadian rhythm dysfunction potentially contributes to opioid addiction. Now, I'm also going to present some fairly heavy molecular and genomic stuff, but I will also try to zoom out to highlight the bigger picture and the implications of some of the data and where that data is bringing us to understand more about the pathology that contributes to opioid use disorder. So I just want to orient us to kind of the major terminology and system that I'll be talking about today. For those who are not familiar, sleep and circadian rhythms are homeostatic processes. They're absolutely critical to our daily functioning. They allow us to adapt to our changing environments, right? And one of the major entraining stimuli that we encounter every day is light and food and potentially exercise and social work schedules, right? And all of these things actually impact our body to synchronize, entrain, and adapt not only our brain, but the rest of our body to those changes in the environment. And we believe that the circadian system is hierarchically organized. So just to give you an example, light comes in through the eye. I don't know if my pointer works. Light comes in through the eye, gets transduced into a number of different chemical signals. Those signals are communicated through the rest of the brain. And then the brain communicates those timing signals through a number of different outputs, changes in body temperature, hormones, changes in sympathetic and parasympathetic nervous system balance, to the rest of the tissues in our body. And this hierarchical system is believed to entrain or synchronize us to these stimuli. Now, the thing that I would also like to point out is that every tissue in the body has what's called a clock. And I'll address this a little bit later in the talk, but every tissue in our body has the endogenous innate ability to keep time without any entraining stimulus, okay? And that's in nearly every cell, including red blood cells. And over the past 10 to 15 years, what's become appreciated is that the desynchrony between our environment, so such as shift work or changes in social schedules, staying up late, having to work late, and so forth, desynchrony between our external environment and our internal environment is a primary contributor to a number of different health-related issues and diseases. The biggest one probably over the past 20 years that has been studied has been cancer. But over the past 10 to 15 years, there's been accumulating evidence in psychiatry that this system is very important for the pathogenesis of a number of psychiatric disorders. So that's kind of where we lie. We've been developing this kind of schematic or working model where we think that sleep and circadian rhythms are essential to a number of different processes, both in the brain, at the circuit level, but also at the gene and protein level, and also at the genomic level. And the interactions between this sleep and circadian impact and the regulation of sleep and circadian rhythms in the context of neurocircuitry and biology definitely seems to contribute to the emergence of certain psychiatric and medical diseases. In our lab, we also consider environmental factors that impact independently brain function and body function at different biological scales. And we always have this kind of tune on how can we potentially drug or target sleep or the clock to improve either current therapies for certain psychiatric disorders, or can we actually create new therapies that might be highly effective for these disorders. Now, as I mentioned at the beginning of the talk, and I probably don't have to say it to this audience, but we've been primarily focused on the relationship with sleep and circadian biology in the context of opioid use disorder. We know that OUD is a chronic relapsing brain disease. It affects millions of people annually. It has a 7 to 10x higher death rate due to a drug overdose compared to 20 years ago. I think this past year was the first year in a number of years that overdose fatalities actually decreased in the US. There are a number of current treatments for people with OUD, but the two mainstay treatments for people with OUD are methadone and buprenorphine. Despite the efficacy, and even if you equate access to methadone and buprenorphine, there is still a very, very high relapse rate. And it's estimated within the first year to 3 years that about 90 to 95% of people actually relapse at some point. Okay, and relapse can look like a number of different things, but we actually see this as kind of an entry point that maybe we could get better. And so this is where the lab has centered itself for several years now. Now, there are a number of what have become canonical symptoms that are associated with acute and protracted withdrawal from opioids. Many of kind of the prior decades of research on opioid dependence, opioid addiction, and opioid withdrawal have really been focused on this acute 24 to 72 hours. And this acute 24 to 72 hours is primarily characterized by these flu-like symptoms, okay? But what we've been really interested in is what happens later. What happens weeks out, months out? And one of the things that has become more and more appreciated, I think, at both the preclinical, the translational, and the clinical level, is that some of the most persistent and severe withdrawal symptoms that last weeks, months, if not years, have to do with these changes in sleep, arousal, and agitation. We know that there's been a number of studies showing kind of an intricate bidirectional relationship between these sleep and circadian changes in the context of opioid use disorder. We know this is bidirectional in the sense that chronic, repeated sleep and circadian disruptions can actually contribute to drug use. What I'm going to be talking about today is mainly the other direction of the relationship, which is how do chronic opioids impact sleep and circadian rhythms that might contribute to craving and relapse. So we do have an interest in looking at the other end where it's vulnerability to, if anybody's interested. But most of this talk is going to focus on the consequences of chronic opioid use. Again, I like to hammer this point, is that some of the recent epi studies that are coming out, and some of the studies that have not yet been published, particularly from groups from West Virginia and Johns Hopkins, estimate that almost 75% of people with opioid use disorder actually reach the clinical threshold for a co-occurring diagnosis of a sleep and circadian disorder. And this is actually very, very different than psychostimulant use disorders like cocaine or methamphetamine. You still get sleep disruption, the persistence, the severity, the impact on craving intensity, all of those things differ between whatever the primary drug is being used. Now, just to kind of prove to you in a more clinical setting, this was a paper that came out from Kelly Dunn and Andrew Hoon's group at Hopkins. Kelly Dunn has since moved. But they created a very small RCT, randomized clinical control trial, with people undergoing a short-term buprenorphine taper. And they gave one group suvorexant, which is a dual orexin receptor antagonist, and another group placebo. And instead of rating objective measures of sleep and craving and so forth, they actually rated subjective measures of sleep and craving and so forth. And it was important because I think the actual patient experience, the subjective feeling that I feel like I'm sleeping better, I am sleeping better, and I have reduced cravings is probably more important than the objective measure. But basically what they showed is that patients going through a buprenorphine taper with the suvorexant. Now, this dual orexin receptor antagonist is meant to impact sleep. It's meant to actually reduce sleep latency and also increase sleep duration, okay? The patients that had both the buprenorphine and the suvorexant actually significantly rated their craving as reduced, and they perceived themselves as sleeping better over those several weeks. At the end, they asked them, did it improve your craving intensity? And the patients with the suvorexant also rated that their intensity of cravings was significantly reduced. So this is a point that our lab has taken to heart, is that we're not necessarily saying sleep and circadian biology is the root cause, right? It is not the root pathology of opioid use disorder. However, use as an adjunctive approach, as an approach to target in the treatment of patients with OUD, can actually improve therapeutic outcome and current efficacy of mainstay treatments such as methadone and buprenorphine. All right. So I'm going to get off my soapbox for a second. And I'm going to describe kind of how we've approached this question. How have we gotten to look at the relationship between sleep, circadian biology, and opioid use disorder? And I have to do one thing before I do that. And I'm sorry that I have to get so molecular. But if you remember one thing from this slide, okay, I mentioned that every tissue in the body has a clock, right? All of our tissues have cells. Every cell in our body has a clock, okay? And that clock is made up of ovals and rectangles, okay? And those ovals and rectangles are genes and proteins that create a feedback loop, all right? This is a classical negative feedback loop. We have stuff that goes up during certain times of day, and we have stuff that goes down at other times of day. The things that go up at certain times of day feed back and shut down the things to put the other things go down at other times of day, okay? And the positive loop is made up of a series of transcription factors. These transcription factors interact with each other. They bind to these fancy enhancer elements in the promoter of the genes, and they drive transcription across the circadian time scale, okay? So we have a peak and trough every 24 hours, okay? Peak, 12 hours, trough at another 12 hours. And some of those genes that are controlled by this positive loop, once they're translated to proteins, feed back to this transcription factor complex and shut it down. Okay, so that's where we get the negative feedback loop, all right? What we've come to appreciate is that every cell in the body expresses this positive and negative loop, but the things that they control, the genes that they control, differ tremendously between tissues and also the phase and the temporal specificity of those genes in that tissue, all right? So clocks, uniform. Every cell of the body, the things that they control across cells and tissues are specific to that cell and tissue. All right, that makes sense. Okay, so we've been trying to look at this in the context of the human brain. All right, and one approach we've been doing is using large-scale, high-throughput multi-omics, and I'll explain what that is for those who don't know in a second, in postmortem brain tissue collected from individuals with and without a disease, in this case, opioid use disorder. And I'm going to be presenting data primarily from the nucleus accumbens, okay? The nucleus accumbens is part of the ventral striatum. We care about the nucleus accumbens because it's involved in a lot of different things, including the pathology and pathogenesis of opioid use disorder. It's involved in reward, motivation, goal-directed behavior, anhedonia, a number of things. But the other thing that the ventral striatum does, the nucleus accumbens does, is that it actually controls and modulates arousal and wakefulness. Okay, so it is an integrative hub for controlling goal-directed behavior, but it also sends outputs to control arousal and wakefulness. So what did we do for the first set of studies? We created a cohort of 20 subjects versus 20 subjects, 20 control subjects, 20 subjects with opioid use disorder. We matched them on a number of different things, including age, PMI, brain pH, RNA integrity, all these things we typically care about in the context of human postmortem brain tissue. We dissected different subregions of the striatum, including the nucleus accumbens, and we threw all that tissue per subject into a blender, and we extracted RNA to do gene expression, all right? Now, there are a couple of things that I would like to point out about this cohort is that all of our control subjects died from natural causes, car accident, gunshot, hit by a car. 18 of the 20 subjects with opioid use disorder, and this is an important point because I'll come back to this at the end of the talk, but majority of the subjects with OUD actually died from acute drug toxicity, overdose, okay? So these are people who had a long-standing clinical diagnosis of OUD. We chose subjects who had a start of diagnosis for at least 4 years. So these are people who are actively using prior to drug overdose for years, and they were probably obviously using prior to that diagnosis, but they also died from acute drug toxicity, okay? So for this cohort, we can't necessarily separate out the long-term effects of opioids on the human brain versus the additive or synergistic effects of acute drug toxicity at the time of death with those chronic effects. Okay, so I say this every time I present this. I don't expect you to read any of those small little labels. But the first thing that we did was we looked at all the differences in gene expression in the nucleus accumbens in people with and without OUD. And I illustrate over there on the left is called a volcano plot. On the x-axis is log 2-fold change, all intents and purposes. That's the degree of change of expression. So if you're on the right of the graph, it's increase. If you're on the left of the graph, it's decrease expression, okay? And then just how the effect size is on the y-axis. So how large is that change of expression, okay? And one thing that you notice very quickly is that we actually have a significant amount of signal, a lot of genes, several hundred that significantly go up, and a little over 1,000 genes that go down. Now, to put this in context, in other human postmortem studies, a lot of the human postmortem work has come out of people with mood disorders or depression or schizophrenia. And differentially expressed signal typically varies, but what we would expect from those types of studies is in the order of dozens, maybe several hundred. So this was actually very surprising to us. We have significant amount of gene changes in the human nucleus accumbens associated with OUD. If you take those top transcripts that are differentially expressed in this heat map, okay, you don't need to know what the transcripts are, but each one of those columns is a subject, and you say, hey, I'm going to cluster all of you guys together based on your gene expression pattern. If the disease, if the OUD was a major effect, what you'd expect is that all the subjects with OUD cluster with OUD, and all the subjects that are control subjects, healthy subjects, otherwise healthy subjects, cluster with healthy subjects, okay? And that's basically what we show, except, so blue is OUD, purple is control, except for these guys right here. And maybe someday Stefan, Dr. Puig and I will actually do a study on this, but these guys have OUD and a comorbid inflammatory disease, okay? Arthritis, something else, chronic pain issue. Many of these genes that we picked up, this isn't really that important, but for those who are interested, a lot of the genes that we picked up are coding genes, but we also have significant amount of long non-coding RNAs too. All right, so we looked at gene expression. There's a lot of stuff changed associated with OUD. We looked at protein expression as well, using shotgun proteomics with mass spec in the same subjects, in the same brain region. And the scale of those expression changes at the protein level were much different. They were much reduced, right? So we had almost 1,300 genes that were differentially expressed. We have on the order of maybe 40-ish, a little over 40 proteins that are differentially expressed. So we wanted to look at what the underlying biology that could actually explain. And again, this is the same type of volcano plot. Increase in red, decrease in blue, and then the effect size on y-axis. But we wanted to look at, try to distill this gene and protein information into something that was tractable, something that we could actually study in the context of the disease. And so we did a number of different things, and I'm not going to bore you with what we did computationally, but we found a number of enriched pathways. So enriched biological pathways that were from the gene and protein data that were significantly changed in the context of OUD and the nucleus accumbens. And this was a bit serendipitous, okay, in the sense that we were hoping that we could target something related to sleep and or circadian biology. But some of the top pathways that popped up at both the gene and protein level had to do with circadian rhythms, all right? So again, we wanted to drill down to see if we could actually capture this temporal variation in the human postmortem brain. Now, how do you do this, right? And so we also do mouse studies. In mice, we can entrain mice to a light-dark cycle. We can sacrifice those animals at different times of day. We can collect the tissue, and we can look at gene and protein expression at those different times of day and recreate rhythmic curves and fit those rhythmic curves through a number of different approaches. to recapture or recapitulate those gene or protein rhythms in the mouse. In human, obviously you can't do that. And so my lab with a couple of other labs have improved upon this approach, which is called time of death analysis, where you essentially use the time of death of each subject, okay, and the date of death of each subject to normalize that time of death to what's called Zeitgeber time, all right? Zeitgeber means time giver, okay? And so ZT0 was normalized to sunrise on the date of the death of that subject, and ZT12, 12 hours later, was normalized to the sunset of that subject, okay? So if you have a bunch of subjects that died around different times of day, and they encompass both your control groups and your disease groups, you can arrange people along this clock. And then you can take each one of those people and align them across this normalized Zeitgeber time. And you can measure the gene or protein expression. And then you can recreate the rhythmic pattern or whatever temporal pattern is using those human subjects. Is everyone on the same page in that one? Okay. So if you do that, you can do a number of different things, but for the sake of simplicity, you can categorize the changes in that temporal variation between the two groups. You can say, oh, in my healthy group, things are rhythmic, and these things lose rhythmicity in my subjects with opioid use disorder. Or you can say, oh, those things are not rhythmic in my healthy group, but they quote unquote gain or become rhythmic in my subjects with opioid use disorder. Now, if you were able to do that, then the heat map should look something like this, okay? So this is a heat map where every row is a gene and every column is a subject, and subjects are ordered by this normalized Zeitgeber time, this normalized ZT. Yellow is increased expression. Blue is decreased expression. And if you were to believe me that we could actually do this or recapitulate or capture some temporal variation in our healthy subjects, what you'd expect to see are these nice blue and yellow waves of changes in increased or decreased expression of those genes as a function of ZT. And that's exactly what we see, okay? When we take those same top 250 genes that are rhythmic in our unaffected subjects, and we plot them in our subjects with opioid use disorder, you can see that that temporal structure completely deteriorates, okay? So these things become arrhythmic or lose rhythmicity, the temporal structure in the context of opioid use disorder. And this is just an example of two genes of actually, what we're doing. Again, x-axis is Zeitgeber time, y-axis is expression. This is a RNA binding protein that loses rhythmicity in subjects with OUD. And this is a microglial gene that actually, quote unquote, gains rhythmicity in OUD. When you do that and you plot all the temporal structure of those genes between the two groups, and you look at when those genes actually peak in expression, right? We have rhythms, we have rhythmic curves that we fit. We've done some statistical hoop jumping. We can normalize and permute that data, and we can say, oh, these genes are significantly peaking at this phase, and these genes are significantly peaking at this other phase. And in our unaffected subjects, what you can see is most of the rhythmic genes that are in the nucleus accumbens in our unaffected subjects actually peak nearly the same time, same phase, okay? So this is what's called a polar plot. It's 360 degrees. 0 would be ZT12, and as you move along the clock, you get to 24 hours. So when you see a trajectory or a vector that goes from the center of a polar plot out to the edge, the length of that vector is the amount, right? The numerical amount of genes that are lying along that vector, and where that vector peaks is the hour of peak, okay? So when you take those genes in opioid use disorder subjects, what you can see is that there are actually a lot more genes that become rhythmic, but the pattern of these genes is nearly completely, well, is totally in antiphase with one another. It's 12 hours apart. All right. So it's changing the entire temporal structure of the gene expression pattern in the nucleus accumbens. And a lot of these genes have to do with synaptic function. Okay. So what did we do? I didn't do this. Dr. Puig did this when she was in the lab at one point. We decided to look at a different biological resolution. So instead of taking the whole nucleus accumbens, throwing it into a blender, grinding it up, and looking at all the things that are changed at the gene and protein level, we wanted to increase that resolution and look specifically at the synapse. So we isolated synapses from subjects with and without opioid use disorder. And then we ran mass spec to do proteomics on those proteins at the synaptic level. And what did we find? Well, you find anything anytime you do these types of studies. But what we found was that there were genes at the synaptic protein level that are up or down regulated. Now, the next thing was not something that we totally expected, and this kind of surprised us. When we did the exact same analysis at the synaptic proteome level, the same exact time of death analysis, and took the top 200 rhythmic proteins and ordered those subjects by ZT, in our healthy subjects, 200 proteins, well, there's actually more than that, but these are the top 200 proteins. A lot of the proteins at the synaptic level actually have a temporal structure of expression in the human brain. Now, this is something that we and others have found in the mouse, okay? When you take those top 200 proteins that are rhythmic in our unaffected subjects and you plot them in OUD, we again lose that structure, all right? These are just some of those proteins that are enriched at the synaptic level. A lot of these proteins, might not be of immediate interest, but we have some proteins that are major players, both in circadian synaptic functions, such as GSK3 beta. But GSK3 beta has also been a failed target of several pharmaceutical companies for treating bipolar disorder and substance use disorders. A lot of these proteins had to do with basic synaptic function. All right. So this is not, some spurious synaptic pathway. This is not some unique synaptic pathway. This seems to be at the root basic function of synaptic processing, because a lot of what we see changed in the pathways of, a lot of what we see enriched pathways that are changed in protein rhythmicity have to do with basic ER function, basic membrane trafficking, okay? When we looked at what's similar or dissimilar between genes and proteins in our unaffected subjects and our subjects with opioid use disorder, now this is just comparing the raw overlap. Are you rhythmic at the gene level? Are you rhythmic at the protein level? This surprised us a ton. But a lot of what we were seeing at the gene and protein level that are rhythmic in both our unaffected subjects and our subjects with opioid use disorder are totally different. So the genes and the proteins and the temporal variation, or at least what we're able to capture in this cohort, which is a limitation, but at least what we're able to capture in this cohort are totally different. This slide, for whatever reason, I've tried to fix several different times, but for whatever reason doesn't show up. So you can just believe me. But a lot of what we saw that was common between the gene and protein across biological levels, again, had to do with basic synaptic signaling, okay? Had to do with basic vesicle-mediated transport, postsynaptic membrane functioning, okay? And we do have specific proteins that exist at the synapse, including GSK3 beta, the solute carrier, and LSAMP, that seem to be viable hits that we want to explore. But this does seem to be a total disruption of this basic synaptic functioning in the context of opioid use disorder. So we drilled down even further. And what we did next is, again, instead of grinding everything up and throwing it into a blender and not being able to separate all those things out, we wanted to look at the single cell level, all right? And we've been doing a significant amount of what's called single cell genomics using 10x Genomics platform, modified 10x Genomics platform. And we did this in a first cohort, again, comparing healthy subjects with opioid use disorder. And we did it in several different brain regions of the striatum. I'm going to present you data on the dorsal striatum, which includes the caudate and the putamen. And we care about the dorsal striatum because it's important for habit formation, compulsion, reward, and may also be important for mood-related craving intensity in those people undergoing treatment for opioid use disorder. So what do? You take the postmortem tissue, you isolate the nuclei, you put those nuclei into little beads, you barcode those beads, you burst nuclei, you sequence those nuclei, and then you can sequence all of those nuclei independently. And then you do a lot of painstaking computation for months, if not years, where things don't always work, to try to differentiate those cell types that you've isolated from the human brain, and then look at what could be changed in those cell types as a function of disease, okay? We published this paper at the beginning of this year in Nature Communications, if anybody's interested. So first thing we did was we wanted to make sure that we were able to identify major classes of cell types in the human striatum that we expect to see, okay? And expect to see meaning all of the historical data from mouse, rat, non-human primate, all of the prior human postmortem gene expression data, not at the single cell level, but at the bulk level. And you take all of this gene expression data and you start to cluster these gene expression patterns to reveal certain cell types, all right? And what we'd expect to see were canonical cell types. So these canonical cell types that express dopamine 1 receptor or the dopamine 2 receptor. But we also captured a lot of lower abundance subpopulations of neurons in our data set, including neurons that express both the dopamine 1 and dopamine 2 receptor, and separate subclasses of interneurons. So the point here is this is a high quality data set where we have a tremendous amount of depth that we can start to dive into at the single cell level in the human brain in the context of opioid use disorder. And we took all of this expression data and we wanted to predict things that could be upstream, things that could regulate genes at the single cell level that are changed in people with opioid use disorder. Okay, so let's say you have all these genes changed in cell A, all of these genes changed in cell B, different genes changed in cell C. We wanted to use cell A, B, and C to see if there was a common upstream transcription factor or common upstream regulators of those gene changes by cell type that was significantly associated with opioid use disorder, okay? And this is a fancy way of saying is differential transcription factor gene regulatory module in OUD, whatever that means. But basically what this means is that on the y-axis, we have the transcription factor, we have the protein. On the x-axis, we have the specific cell type, okay? Anywhere you see a box that is bolded or outlined in black, those are significantly associated with opioid use disorder. And they're significantly changed in those specific cell types, okay? So the simplest way, they're explaining a ton of the gene expression variants at the single cell level that's associated with disease, all right? And we predicted a lot of transcription factors that go up or down as a function of OUD and specific cell types, including this protein transcription factor called clock. Now, clock, if you remember from the positive negative feedback loop, clock is part of that positive loop, okay? It's a transcription factor that drives gene expression at different times of day, all right? However, anytime you do genomics and anytime you rely on other people's annotation including public databases, if you don't know anything about the biology underlying those annotations of public databases, it limits you. We know that clock is actually not that highly expressed in the striatum, from mouse all the way to human, okay? But there is another transcription factor that is highly expressed, and it's its homolog, which also binds the same protein clock does, and is also part of the circadian positive loop, and that's called MPAS2. And this is just a paper from Garcia et al. In Science, now 24 years ago, where they actually show that this MPAS2 transcription factor, this black hole, is the mouse nucleus accumbens. Okay, so we looked at MPAS2 expression in different neural subpopulations of the striatum and individuals with OUD. And these are horizontal violin plots, where on the y-axis you have cell type, on the x-axis you have relative expression. The higher the bubble, the bigger the bubble, means more expression, okay? As you move from left to right, you get more specific in terms of the cell type and the disease interaction, okay? So we looked at MPAS2 expression. It seems to be primarily enriched in medium spiny neurons, which were these D1, D2 neurons that I mentioned earlier, okay? When you look at the expression of MPAS2 in these medium spiny neurons, the red is opioid use disorder, gray is control. This bubble is less surface area than this bubble, meaning reduced expression. So we have reduced expression of MPAS2 in OUD subjects. And then when you further divide those MSNs into different subpopulations, we show that MPAS2 is actually less expressed in OUD in D2 and D1 striosome neurons. Okay, so we've gone from bulk data and used our single cell data to somewhat non-biasedly predict a circadian transcription factor regulatory network that's potentially involved in opioid use disorder. Okay, and we've drilled down to a particular cell type. Okay, we think it's changed in D1 and D2 neurons of the human striatum. Why do we care about D1 and D2 neurons in the nucleus accumbens? And I'm not going to go over all of this, but D1 and D2 neurons in the nucleus accumbens send projections to different brain areas, including the midbrain, the ventral tegmental area, the ventral pallidum, and so forth. But the output of these neurons differentially controls drug-related behavior, differentially controls motivated and goal-directed behavior. Now, I'm summing up a significant amount of research, and there's a tremendous amount of nuance to this research, both in human and animal, but for all intents and purposes, activation of D1 neurons in the striatum tends to promote goal-seeking behavior, tends to promote reward-related behavior, okay? Activation of D2 neurons tends to antagonize that behavior, right? So you can think of this as a go signal versus a stop signal, all right? So we know that MPAS2 is changed in both. We know it's enriched in the nucleus accumbens of mice. We know that it's predicting a transcription factor regulatory network in D1 neurons, particularly in subjects with opioid use disorder. So we wanted to bring this to the mouse, all right? We wanted to explore if MPAS2 is doing anything in the mouse. And we did a very simple study, very crude study. You take male and female mice, you inject them with saline or acute single injection of fentanyl or repeated daily injection of fentanyl over the course of 7 days. You sacrifice them, you take out their nucleus accumbens, and you look at the expression of MPAS2, and we also looked at the expression of the homolog clock. And what we showed is if we were able to corroborate some of the findings that we were at least seeing in human, was that we predicted a decrease in MPAS2 expression, okay? And that's what these orange bars are. So with both acute and repeated fentanyl in both male and female mice, you see a significant reduction of MPAS2 in the nucleus accumbens. Now, this is not cell type specific, all right? This is just at the bulk level, okay? We wanted to go back to the human data because we wanted another piece of evidence that we should be pursuing MPAS2 in the context of opioids, right? So remember when I told you we can categorize change in temporal structure or loss of rhythmicity or gain of rhythmicity of genes between healthy subjects and subjects with opioid use disorder. So we went back and we categorize all the things that lost rhythmicity between our healthy subjects and our subjects with OUD. We had about 406 transcripts. And we did the same type of transcription factor analysis. Are there things upstream that could be regulating these genes, more likely than not, right, that could explain this level of loss of rhythmicity? And we identified two. And the two we found were MPAS2 and HNF4A. Okay, so now we have another piece of evidence in the human brain, right, in addition to our single cell data, that MPAS2 is doing something in the striatum, and it's changed somehow, some way, in the context of chronic opioid use. And we might want to look at it. All right. So we turned to a mouse that we actually got from David Weaver way before that I was even contemplating coming to UMass or even moving back to New England. But these mice are globally MPAS2 deficient animals. What does that mean? These animals are not a knockout. We just deleted a part of the protein, and it renders the protein, the transcription activity of that protein, impaired. Right? So these are not a knockout, but they have the transcription factor, the activity, the DNA binding activity of that transcription factor removed. And we wanted to look at whether the behavior in these animals, when they didn't have transcriptionally functional MPAS2, regulated any opioid reward-related behaviors. And so we started with a fairly simple assay called condition place preference, which is a contextual learning assay where the animal is placed on either side of a 3-compartment box. The two far sides of that box differ in its appearance. The floor might differ, wire versus bar, okay? So the animal can not only feel, but see the contextual differences. And on one side, you give them a saline injection, which is relatively inert, or on the other side, you give them fentanyl. Now, most animals, if they've developed a conditioned response, right, the unconditioned stimulus becomes the CR, is that they prefer to stay on the side that they got the drug. Now, this is classical condition reward, right? They've associated the context with where they administer the drug. And we did this in globally MPAS2 deficient animals, both males and females. Wild type is the neutral colors, and MPAS2 deficient are the blue or purple, males or females, across different doses. And what you can see is that this MPAS2 deficiency actually leads to an increase in condition rewards, both in males across dose and in females across dose. So this was exciting to us because, and at the time, Dr. Puga had done some other stuff with pain with these animals, but this was super exciting for us because it got us more interested in the fact that, we have converging evidence across humans and mice that in a particular brain region, maybe in a particular cell type, that regulates our response or the animal's response to opioids, okay? Now, one of the things we wanted to do was to develop a way where we could create an environment where the mouse could volitionally control their administration of the drug. And we had the question, does MPAS2 drive the volitional control? Does it drive drug-seeking behavior of fentanyl? Okay. And we wanted to actually develop an approach where we were doing oral self-administration of fentanyl because what's become quite apparent, and we're probably in the 4th or 5th wave now, but what became quite apparent around 2015 was that synthetic opioids were hitting the mainstream market very, very hard. They're very cheap. They're readily available. You can transport them very easily. But because of their high potency, you don't have to inject. You can snort, smoke, or orally consume. And at the time in 2015, actually, most people were using some type of compressed fentanyl pill, fentanyl lozenges, or tab, like LSD or acid. And so we wanted to create an animal model where the animals were actually trained to press a lever to gain access to fentanyl-containing solution that they could drink. All right. So the animal is pressed to press an active lever. If they press the active lever versus the inactive lever, it dispenses fentanyl into a dipper. The animal then has to learn to go over to that dipper, get interested in whatever's in that dipper, consume that fentanyl-containing solution, and associate whatever I just drank, I gained access to because I pressed the active lever, okay? And once you train the animals to do this, you can gain a lot of different insights into the behavioral sequelae that are associated with drug seeking and motivation. And we did this both in male and female wild type and MPAS2 deficient animals. The most consistent effect, for the sake of time, I'll simplify this. For the most consistent effect, it was in this NPAS2 deficient females. This was actually the number of rewards given. So the number of times they pressed the active lever and it dispensed a fentanyl-containing solution. But we could also measure the amount of fentanyl consumed in that dipper after the session, which is an important measurement because if they want it more, they should be consuming it more. And female MPAS2 deficient animals tend to have consistently higher reward administration and fentanyl consumption across 2 weeks of this active lever, inactive lever operant self-administration paradigm. But we didn't stop. We wanted to look at whether this could be explained by a cell type specific mechanism, right? Because we had some cell type specific data from human. We had some other cell type specific data from our animals, and we mapped MPAS2 expression across the entire mouse striatum and specific cell types. And what we show is that most of the MPAS2 expression is in D1 or dopamine receptor expressing 1 neurons. Those were the neurons that I said those were the go signal, right? They're driving the motivated behavior. So our hypothesis was, well, maybe opioids impact these D1 neurons. MPAS2 somehow loses its normal function. And this loss of normal function contributes to this increase in this opioid reward-related behavior. So we did this several different ways. I don't know why these are showing up blurry, but we created a virus, the short hairpin RNA, where we could actually knock down MPAS2 in D1 versus D2 neurons. And we again did this condition place preference assay. And we did this in a Cree-dependent way. So we could knock down MPAS2 only in dopamine receptor 1 expressing neurons versus 2 neurons. And we did this both in males and females. As you can see in both males and females, knockdown of MPAS2 recapitulates the increase or the augmentation of fentanyl condition place preference that we saw in the global MPAS2 deficient animals. So it seems to be MPAS2 acting in D1 neurons, not D2 neurons. And this is kind of our working model where this loss of MPAS2 function, for whatever reason, which is what we're exploring more now, seems to drive this reward motivated behavior. We have a number of other studies that we've done with this, including MPAS2 deficiency impact on sleep and arousal, particularly sleep loss during opioid withdrawal. And we created some new mice where we were actually able to CRISPR out MPAS2 specifically in D1 neurons and subregions of the striatum to also look at fentanyl and sleep impact. So with an orientation towards trying to figure out, is MPAS2 targetable? Like, can we target through other means besides virus? Are there pharmacologies, compounds that we can use to target MPAS2? I did this experiment a really long time ago when I was, well, doesn't matter. So we wanted a pharmacology that drove MPAS2 expression, right? MPAS2 is down impacted by opioids. So we settled on this, what's called a ROR agonist, this ROR alpha gamma agonist that actually drives MPAS2 expression. We gave it to mice, and we thought if this is definitely, somehow involved in how the animal responds to fentanyl, then we should be able to drive MPAS2 expression and gradually decrease the behavioral response of that animal to fentanyl, okay? And we gave the animal the compound, this SR1078. We looked at MPAS2 expression in Bliss Cummins. It was significantly increased. So at least it was going in the direction we expected it to go. This is just the vehicle control group. And this is a pretty dirty experiment. It's clumped over 2 weeks. But what you can see is animals with the compound over the 3rd week of self-administration of fentanyl actually reduced their overall responding. All right. And we're pretty excited about this. I have a postdoc who's looking at another orphan receptor that might be upstream in this context. So just to quickly review, altered diurnal rhythms and transcription expression in opioid use disorder seems to be related to synaptic signaling in OUD. These synaptic signaling seems to be rhythmic, and the loss of this rhythmicity within the nucleus accumbens seems to be related to opioid use disorder. We have a number of other different projects in the lab, and I mostly use this as time to kind of just advertise what's going on in the lab. We have a number of different things, but our approach to opioid use disorder is multi-pronged, and it's mostly at the genomic and molecular level. And we've just gotten 2 new R01s this year to explore the interactions of some of the stuff that I've talked about in the context of HIV and major depressive disorder in people with opioid use disorder, and also how some of these other molecular mechanisms that I wasn't able to touch upon in the nucleus accumbens might be key regulators of opioid craving and relapse. And with that, I won't go into this, but with that, I'd like to thank my lab. Most of the people are here. I don't know why the link's not working, but that's my lab up until Matt in the hat. He didn't choose my lab. He chose Dr. Puig's lab after he rotated with me. So he's okay. He's all right. And I'd like to also thank past members of lab, including Kelly, Barco, Bedoy Fan, Stephanie Puig, Maddie Ray, and Micah Shelton, who are all in my labs at different stages, either between Pitt and BU, and particularly collaborators Jill Glosher, Matt McDonald, and Marianne Sine, and Andreas Fenning, who have been absolutely instrumental in the human postmortem work. And of course, want to thank the current funding and the past funding. Thank you.
I'll get you.
So that was wonderful. Really, really interesting work. One of the things about individuals with opioid use disorders, there's a very high rate of PTSD. And I heard you're going to be exploring depression and OUD comorbidity. And then one really interesting thing about individuals with PTSD is they don't sleep for love nor money. So I'm just sort of wondering if there's some of this that might be applicable to that population, or if it could be moderating your results in some way if somebody has PTSD comorbidity.
Yeah, so I didn't get too detailed into this, but the way we select our individuals for cohorts for these types of studies, there's a set of inclusionary and exclusionary criteria. And these are not just pulling data from a medical record. The subjects that go into the brain bank go through the family or next of kin, goes through a full 12-month interview with the neuropsychologist, the psychologist, and the social workers. All of that data is validated, curated, cross-checked between the medical and clinical records and the interview information. All of that data is then scored either numerically or annotated with terms. So in everything we've done to this point, all of our subjects, the exclusionary criteria, PTSD is one of them, okay? However, in our new cohorts with major depressive disorder, it's a little more complicated. We've excluded people with PTSD, but we've included people with opioid-induced depression versus people who have had depression and have now been diagnosed with opioid use disorder. And that creates more mud that we're going to have to deal with it statistically. But we think we can do that because the scale for which we're doing that. The next project is several hundred subjects per group. Yeah.
Thank you. That was really cool. But you have a knack for making things sound like I could almost understand them. I can't, but it's almost, I can feel it. It's right out there. So you went through a couple of slides, one where you showed gene products and a chunk of them were immune related.
Yeah.
And then another one where you looked at the difference in I think it was cell membrane proteins in the two. And I noticed, for instance, Iglon-5 lit up, which has an autoimmune encephalitis associated with it. And I know it's complicated I'm always guilty of taking like things you hear about in a mouse and thinking you can apply that in people and speculating wildly. But the question is, does the nucleus accumbens, the function of the opioid system in withdrawal, et cetera, and its relation to the immune system mean that people with opioid use disorder have a differential risk of autoimmune encephalitis and cancer? And if so, way in the future, first of all, is sleep a treatment for cancer? Okay, you don't have to worry about it. And is NPAS-2 modulation, is that a way that you can impact the immune effects or the anti-immune effects of opioid use disorder? Because it seems like this could be, I mean, it's wild in the future, but it seems like it's relevant.
Yeah, it's definitely relevant. The next time I present this, I'll definitely remove immune from any of the slides. So I'm just kidding. So a postdoc who's actually sitting in front of you, Samara, and a graduate student who's been in the lab, who followed me from BU to here, Mag, has been exploring this intersection with immunology, neuroinflammation, pro-inflammatory signals in the brain that could be contributing to a whole host of things that we think we're measuring in the context of OUD. Most of that is done in the mouse, right? Because it's a tractable model, we can do a lot of different things. What I didn't touch upon, and I tabled this a number of years ago, most of what MPAS-2 actually regulates at the transcriptional level in the nucleus accumbens has to do with immune function. And this was a little bit confusing at the time, right? Because we showed that MPAS-2 was preferentially enriched in neurons. And at the time, I would present that data and be like, oh, neurons don't have an immune response, right? Everything was microglia, astrocytic, right? Well, that's not true. So I also have data to show that MPAS-2 heterodimerizes with one of the major pro-inflammatory canonical markers, NF-kappa B, in neurons in response to cocaine and opioids. And it's actually a neuronal immune response. This is my hypothesis. It's a neuronal immune response that is secreted and actually recruits the microglia and astrocytic response to where those neurons are located. So we in the lab have different ways that we're exploring that. We're exploring that in the context of can we ramp down the immune response during withdrawal, see if we can improve sleep. Can we target specifically TLR7, TLR4 mechanisms at the neuronal level to reduce the impact of fentanyl on physical dependence and sleep measures during that withdrawal period? One thing that is absolutely uniform, at least in the mouse data, which I find super intriguing, is that we've been doing cell culture, primary cell culture data with microglia. You take microglia from a mouse pup, you grow them in a dish, and then you can do stuff with them, right? You can expose them to fentanyl, other things. In vivo, if you give animals opioids and you sack those animals and you look at the inflammatory response while the animal's on opioids, it looks very much like controls, okay? When you remove opioids, it goes through the roof. And we see the exact same thing during withdrawal. And we see it in the striatum and we see it in the prefrontal cortex. And we see it in the microglia culture. So it seems to be like opioids on board is doing something to modulate that immune response in the brain. As soon as you remove those opioids, those cells, for whatever reason, immediately ramp up their pro-inflammatory response. And we think that's definitely important for a number of things that we're studying. MPAS 2 might be involved. Yeah. Any other questions?
Yeah, we have time for one more.
Thank you so much for your talk. I learned a lot. I just wanted to know if after like so long on like methadone or buprenorphine, if you ever see like the genes actually start to return back to a baseline, or if adding those treatments actually just keep them kind of dysregulated, and if that could predict like risk of like relapse
Yeah.
if they get back to normal.
Yeah. I promise I'll keep this super short. Because this is something that we're really interested in, right? We're interested in those people that have some set of resilience factors, biological, environmental, social, whatever it is. It's a mixture of any that promote remission, right? And we actually have data to do that in cohorts. So we did sequencing where we could compare acute drug effects versus chronic drug effects versus people who were in clinical remission at the time of death and for months. And had no opioids on board, had not used alcohol, no smoking, nothing. And what you could do is you can map out these different curves, right? So you can imagine these are the molecular changes, arbitrary molecular changes on the Y. These are our different groups. OUD positive, opioid at time of death, OUD negative, no opioids at time of death, remission, right? And our hypothesis would be exactly that. Genes that change as a function of using opioids, what happens during remission? And I can tell you that is super cool. So I don't expect you to read all of this, but you can categorize these gene changes by group. And probably the coolest group is this remission specific effect up here. So this is dorsolateral prefrontal cortex. We all care about the prefrontal cortex, we think. And these remission specific effects. So these are genes that aren't changed significantly between subjects who have a diagnosis of OUD with and without opioids at the time of death. But all of a sudden, they go up or down when the subject reaches remission, right? And these subjects haven't used in months. Several of the subjects have been years, okay? But we can start to map out and categorize these gene changes. What's persistent during withdrawal, what's acute, what's lifetime drug effect, right? What stays up, what stays down as a function of all of these things. The other thing you mentioned about methadone and buprenorphine. So a couple of people in the lab are doing studies with methadone and buprenorphine, particularly in the context of sleep and gene changes. And buprenorphine actually probably makes sleep worse. And the clinical literature is catching up with the animal literature. And so I do think these adjunctive therapies, just to come full circle, I think these adjunctive therapies like Ciprexin or anything or other things could actually improve treatment outcome or treatment efficacy with people on these mainstay treatments. Yeah.
Well, thank you very much. If people have more questions, he is on our faculty and is available if you walk over to the lab.